Hello, Medical Sciences Made Easy by Naphtali Muhumuza. And this session we want to continue talking about Denovo synthesis. In part 1, we have seen where it occurs, we have seen what triggers it, and we have seen the first 2 steps. That is transfer of acetyl-CoA from the mitochondria to the cytosol. And we also saw how this acetyl-CoA is converted to malonyl-CoA, which is a 3-carbon. And we said that this is a rate limiting step, and it is where we see, it is the precursor and it is a rate limiting step. So if whenever I want to limit fatty acid synthesis, I can limit using step number 2. And we have gone ahead to look at the elongation step, whereby it involves fatty acid synthase, and this fatty acid synthase, we said it is a multi-enzyme containing of 7 enzymes, which catalyze the elongation stop of fatty acid, whereby we listed them down here, whereby we have acetyl-CoA, transilase, malonyl-CoA, 3-ketoacyl synthase, 3-ketoacyl reductase, then we have hydratase, enoyl reductase, and thioesterase. So let us see how they get involved in the synthesis of the elongation step. And as we see that we have our fatty acid, this is our fatty acid synthase. This is our fatty acid synthase, which has 2 arms, that is one cysteine group of the ketoacyl synthase, and the ACP having pan-phosphate panthene, which is always carrying the ACP. And we said ACP is acyl carrier protein. So we are going to see how they play a big role. So this fatty acid synthase enzyme is going to, we have an enzyme known as, this enzyme is known as acetyl-CoA transacylase. So this enzyme is the one that is going to catalyze the transfer of acetyl group to the cysteine arm of the fatty acid synthase. So we see it transferring the acetyl-CoA. So it transfers the acetyl coenzyme A to the cysteine group of the fatty acid synthase. So we see it being added at this stage. And this reaction is being catalyzed by acetyl-CoA transilase. And after transferring it at the cysteine portion, we see another enzyme coming into play. And this enzyme is known as malonyl. It is known as malonyl-CoA transacylase enzyme, which is going to transfer the malonyl coenzyme A at the pan site of the ACP. Because I told you this one contains the acyl carrier protein. So the malonyl is transferred at the pan, which is a 3-carbon. So we see at pan side, we are adding malonyl-CoA, which is carbon number 1, number 2, and number 3. So it is added on the pan sulfhydryl group of the ACP. So this enzyme is the one that carries this malonyl-CoA. And after adding malonyl-CoA, this was step number 1, step number 2. At step number 3, we see the condensation. We are going to condense the acetyl-CoA at the cysteine part with the malonyl-CoA at the pan ACP portion. So we are going to combine them, whereby this one, this functional group is going to join at carbon number 2. And when we join it, we cleave off the carbon dioxide. There is cleavage of the carbon dioxide. And we are able to form this compound known as keto keto, because we have introduced another, we have added a keto at position number 3. This is position number 1, position number 2, number 3, and carbon number 4. So at carbon number 3, we have added a ketone group. That's why we are calling it ketoacyl enzyme. And this step is catalyzed by an enzyme known as ketoacyl synthase. Ketoacyl CoA synthase. And this ketoacyl CoA synthase enzyme is catalyzing the condensation or the joining of the acetyl CoA together with the malonyl with loss of 1 carbon. So we are 4, that's why we are forming a 4. Because we had a 2 carbon, if we were not losing a carbon dioxide, we would have 5. But we have 4 because we have lost a carbon dioxide by the process known as decarboxylation. So it catalyzes the joining of these 2 compounds, that is acetyl CoA and malonyl CoA, with loss of a carbon dioxide. And that is step number 3, forming ketoacyl enzyme. And after knowing this, that's why we call it 3 ketoacyl synthase. It is synthesizing a ketone group at position number 3 with loss of the carbon dioxide. After seeing this step, we are going to see ketoacyl enzyme being reduced. This step we are saying reduction. This step here, it is a reduction step. I don't know how I'm going to show it, whereby before we form this compound, we are going to see that to convert a ketoacyl CoA to 3-hydroxyacyl CoA enzyme, we need to reduce. There is a reduction step whereby it involves NADP H, which is got from conversion of malate to pyruvate. We saw there was production of NADPH by the action of malic enzyme, which we are feeding in this step of reduction. And there is another NADPH produced during hexose monophosphate shunt. So we try those NADPH to reduce ketoacyl CoA to 3-hydroxyacyl CoA. And this step, when we see the NADPH being oxidized to NADP class. And the enzyme that catalyzes this conversion of ketoacyl CoA to 3-hydroxyacyl CoA, it is known as 3-ketoacyl reductase. So 3-ketoacyl reductase enzyme, in the presence of NADPH, it reduces ketoacyl CoA enzyme to 3-hydroxyacyl coenzyme A enzyme. And this one involves oxidation of NADPH, which is got from conversion of malate to pyruvate by malic enzyme, and also from hexose monophosphate shunt during carbohydrate metabolism. So we see this ketoacyl reductase is adding a hydrogen. The hydrogen cleaved from NADPH is added at carbon number 3 on the keto group on carbon number 3, forming the hydroxyl group at carbon number 3. That's why we call it 3-hydroxyacyl CoA enzyme. So after forming that hydroxyl group at carbon number 3 by reduction of NADPH, we see this being lost. It undergoes hydratation reaction, whereby there is dehydration or removal of water to form an eno group. So there is hydration process, dehydration, whereby this dehydration, we are removing water from 3-hydroxy to form enoyl. And this process of removing water is catalyzed by enoyl reductase. You see enoyl, no, it is catalyzed by hydratase enzyme. So hydratase enzyme cleaves off water from 3-hydroxyacyl enzyme to form 2,3-unsaturated acyl-CoA. How is it formed? Is that when we remove this hydroxyl group at carbon number 3, we are going to form an eno group. We form an eno group between carbon number 2 and carbon number 3. And this process or this formation is catalyzed by hydratase enzyme. So hydratase enzyme is the one that catalyzes that removal of water to form 2,3-unsaturated acyl enzyme. And this enoyl group which is formed at this point is going to be reduced. It's going to be reduced, and still here we need NADP H, and being oxidized to NADP plus, whereby the hydrogen is reducing by removing this double bond to form a saturated compound which is known as acyl. This one is known as acyl enzyme. And this process requires an enzyme known as enoyl reductase. It's catalyzed by enoyl reductase enzyme. So we are seeing that this enoyl group formed between carbon 2 and 3 is reduced, or it is removed by addition of hydrogen, whereby we remove this double bond to form a saturated compound of acyl enzyme, catalyzed by enoyl reductase because it's a reduction reaction. And finally, after forming this 4 carbon 1, 2, 3, and 4 hydrocarbon chain on the pan site of ACP, we are going to transfer it to the cysteine state. So we transfer it from pan to the cysteine so that we can free the pan, ACP pan site, so that we can allow more malonyl-CoA to come and bind. So we are going to see this is why we are calling it transferring just. We are transferring the 4 carbon hydrocarbon chain formed here from the pan of the ACP to the cysteine, so that we can free the space for another conversion of malonyl coenzyme A. So after transferring it, it will undergo from 2 to 7 cycles until we form a 16 carbon compound of palmitic acid. And when palmitic acid is formed at the cysteine part of the fatty acid synthase, we shall cleave it. We need an enzyme that can cleave it to free this palmitic acid. So we are going to see the cleavage. We are going to cleave the palmitic acid formed after several cycles. What are we meaning by several cycles? Is that after forming this 4 carbon compound, it is undergoing to add by freeing the pan site. We bring malonyl-CoA, we pass through the same processes, we form another 4, we bring another malonyl-CoA. It passes through the same process until 7 cycles when we form a 16 carbon. And when we form this 16 carbon known as palmitic acid, which is a 16 carbon fatty acid, saturated fatty acid, it is formed by cleavage. It is formed whereby the enzyme known as thioestylase, or you can call it deacylase. Deacylase meaning it is removing the acyl group from the fatty acid synthase, and we come and cleave it off after forming a long chain, and we get our palmitic acid, which can be used for formation of other long chain fatty acid. And this is the elongation step, which we can talk about during de novo synthesis of fatty acids. So after forming our palmitic acid, and we can use it to form other long-chain fatty acids, we want to see how this de novo synthesis is regulated. So another step is we can look at regulation. Regulation of fatty acid synthesis. How is it regulated in the body? And it is regulated in 3 steps, whereby number 1, it is hormonal. We have hormonal regulation. Number 2, we have allosteric regulation. And number 3, we have nutritional, according to the nutritional requirements regulation. Whereby under hormonal, we have enzymes like glucagon and epinephrine. Glucagon and epinephrine, they stimulate this fatty acid synthesis. Whereas enzyme like insulin, hormone like insulin, not an enzyme, hormone like insulin, for it inhibits lipogenesis, but these ones stimulate lipogenesis. So that is hormonal control. And which enzyme are we controlling? We are controlling the rate limiting step, which is the conversion of acetyl-CoA to malonyl-CoA by an enzyme known as acetyl-CoA synthase, which is the rate limiting step. So we are seeing glucagon and epinephrine can stimulate acetyl-CoA synthase, whereas insulin inhibits acetyl-CoA synthase. Then allosterically, we see this one being stimulated by increased levels of citrate, as we saw that acetyl-CoA is converted to citrate. Increased levels of citrate stimulate, whereas increased levels of palmitoyl-CoA, enzyme A inhibits. Because we have seen this is the product we are forming. So when we have much of the product, it exerts a negative feedback, whereas much citrate exerts a positive feedback by activating more synthesis of acetyl-CoA synthase, whereas much levels of palmitoyl-CoA inhibits or decreases synthesis of acetyl-CoA synthase. What we are regulating is an enzyme known as, all these things are regulating acetyl-CoA synthase, which is acetyl-CoA carboxylase. It is not synthase, sorry, it is carboxylase. It is the one we are regulating, which is the rate limiting step, whereby we have seen glucagon and epinephrine stimulating this enzyme, insulin inhibiting it. Then we have seen high levels of citrate stimulating more production of acetyl-CoA carboxylase, whereas palmitoyl inhibiting the synthesis of acetyl-CoA carboxylase. Then nutritional, under nutritional, we see increased carbohydrate diet, a diet with high carbohydrate, increased carbohydrate diet, and low fat. These two stimulate the synthesis of acetyl-coenzyme A carboxylase, whereas decreased levels of carbohydrate meal and increase in fat diet full of fat, these two inhibit. They inhibit the synthesis, or they decrease the synthesis of acetyl-coenzyme carboxylase. Because we are saying that the regulation, we target the rate limiting step, which is conversion of acetyl to malonyl by acetyl-CoA carboxylase. And these are the 3 ways we can regulate this process. So after forming our fatty acid, we can use it. We can use this fatty acid we have formed to synthesize the triglycerides. You can use it to synthesize triglycerides. And how do we synthesize triglyceride using so we have formed a free fatty acid, a fatty acid. We have the fatty acid which is formed during Denovo synthesis. We can use it for synthesis. So the heading here is biosynthesis of triacylglycerol. And this one occurs in the liver, majorly in the liver and adipose, whereby in the liver cytosol of the liver, we see the fatty acid combining. It undergoes activation. It is activated to acyl-CoA, and this activated fatty acid is the one that combines with glycerolaldehyde, glycerol 3-phosphate to form phosphatidic acid. We form phosphatidic acid, and this phosphatidic acid formed, we remove the phosphate group. It is cleavage of the phosphate, and we form DAG. And the DAG is known as diacylglycerol. And this process is catalyzed by phosphatase enzyme. This one is catalyzed by acyltransferase enzyme. This is the enzyme that is catalyzing this reaction. This one is the one that catalyzes this reaction. And after forming diacylglycerol, we come and we add another activated free fatty acid. And these two, whereby when we combine DAG with a free fatty acid which is activated, we are able to form triacylglycerol, occurring majorly in the liver cells, cytosol of the liver cells. That the fatty acid is formed here, we can use them to synthesize triglycerides in the body. Thank you so much. We have seen how to synthesize these fatty acids by Denovo, whereby they have undergone series of reactions until we formed this 4-carbon compound, which is transferred to the cysteine, and it undergoes 2 to 7 cycles by bringing a malonyl-CoA, until we form the 16-carbon of palmitic acid, which can be used to form long-chain fatty acids. And it is cleaved off by thioesterase, or an enzyme known as the acylase enzyme. And we have seen how it is regulated, whereby it is regulated at the rate-limiting step. Don't forget the rate-limiting step, which is the conversion of acetyl-CoA to malonyl-CoA in the cytosol. Whereby we can see that it is regulated by hormones, allosterically by the products, and then nutritional requirements within the body. Thank you so much for listening until the end. Always remember to subscribe and share your comments. Thank you so much.